We talk about mitochondrial diseases. This is the tale of two genomes because the mitochondrial products of nuclear and mitochondrial DNA. So this is an image here, an electron, scanning electron micrograph of mitochondria. And they look like these kidney bean-shaped structures. That's the way they look in the textbooks, but they're really much more dynamic than that. And the middle image, middle panel is a series of muscle biopsy sections. The top one here shows a Mori trichrome stain. And normally, muscle fibers have this bluish-green appearance, but you see some other fibers that look very abnormal. Anyone know what they are? One of the hallmarks of mitochondrial disease? Anybody want to guess? Which of the abnormal fibers? This one? Ragged red fibers, yes, right, so good. I'm glad that you've heard of ragged red fibers, okay. So that's what we often look for in muscle biopsies. And when we stain these muscle fibers with a histochemical stain for complex 2, you'll see that they're blue, dark blue, and we call these ragged blue fibers, and I'll explain why they're blue and why they're dark. And if you do serial sections of the same muscle sample, you'll see that those ragged blue fibers are often deficient in complex 4. This is the chemical stain for complex 4, also known as cytochrome oxidase. So I'll explain why you get this pattern in a little bit. And this is what often results in the patients. They often have weakness of the extraocular muscle, so they often have ptosis and ophthalmoparesis, inability to move their eyes. So that's one of the points. So if you remember those images, then you will know quite a bit about mitochondrial diseases. All right, so mitochondria came from bacteria that developed a symbiotic relationship with cells about 2 billion years ago. They started that as protobacteria, probably rickettsia-like bacteria, and they brought with them the capacity to make ATP, energy of the cell, through a process known as oxidative phosphorylation. So this is the cartoon version of the mitochondria. In addition, so these are the enzymes, complexes 1, 2, 3, and 4, which shuttle electrons, which are basically derived from glucose, which is broken down to pyruvate, and fatty acids, which is broken down through the beta oxidation cycle, and it generates acetyl-CoA, which then goes through the Krebs cycle and generates these reducing electrons, which flow through complexes 1, 2, 3, and 4, and that's used to drive proton pumping across the mitochondrial inner membrane. Then that proton gradient is used to drive ATP synthesis through complex 5. So in addition to making ATP, the ability to make ATP, mitochondria brought in their own DNA, this tiny piece of DNA, which is only 16,500 bases in length, which is tiny compared to the nuclear genome, which is about 3 billion bases in length. But that tiny genome is essential for life. It encodes 37 genes, and 13 of those genes encode polypeptides with complexes 1, 3, 4, and 5. Complex 2 here is encoded entirely by nuclear DNA, which is shown by these ovals here. So that's why if you have a defective mitochondrial DNA, specifically if you have a defective mitochondrial protein synthesis or translation, you're going to affect complexes 1, 3, 4, and 5, but complex 2 will be spared. So when you do a stain for mitochondria using a stain that detects complex 2 activity, you'll see that often it's normal or elevated, whereas these other enzyme complexes like complex 4 will be decreased. That's why you see that pattern that I showed you in the first slide. So you need both mitochondrial and nuclear DNA to make this oxidative phosphorylation process work. Okay, this is the cartoon version of that mitochondrial genome, 16,500 bases in length, 37 genes. So 13 encode polypeptides, 22 encode transfer RNAs, and 2 encode ribosomal RNAs. So these genes basically encode either structural components of the respiratory chain, and oxidative phosphorylation, or machinery required to make proteins within mitochondria. That mitochondrial DNA has evolved over these last 2 billion years, and in humans over the last 100 or 200,000 years. And there are differences in the mitochondrial DNA over evolution of humans. So there's trace back the origin of the human species to mitochondrial Eve here in Africa. But it's, you can see that the human population migrated out, the mitochondrial DNA evolved over time. So I told you a little bit of background about mitochondria, the biochemistry. So how does that translate into diseases? Well, mitochondria are essential for making energy, so they're essential for virtually every organ in the body. So mitochondrial diseases with defects of ATP synthesis affect virtually every organ in the body, but brain and muscle are particularly vulnerable. So we often call these diseases encephalomyopathies, but they're often multisystemic. So the brain can be affected in many ways. You can have seizures, movement disorders, strokes, dementia, particularly young people. Muscle is affected. These patients often have weakness. Because they can't make ATP, they often complain of exercise intolerance. They poop out in proportion to their weakness. Another muscle that's frequently affected is the heart. And these patients often have cardiomyopathy. Often this cardiomyopathy is hypertrophic. This is often the case with genetic cardiomyopathy. So different from your typical dilated cardiomyopathy, which is much more common. These mitochondrial patients have hypertrophic cardiomyopathies. They can also have heart conduction blocks, pre-excitation syndrome, or Parkinson-White syndrome. As I told you before, another muscle that's frequently affected is the extraocular muscle. So these muscles are full of mitochondria. If you take a normal extraocular muscle and you section it and stain it for mitochondria, you'll see that they're full of mitochondria. We're constantly moving our eyes, and so as a consequence, when we have a defect in mitochondria, the eye movements can be affected. Patients have ptosis and droopy eyelids and inability to look the eyes. The liver is frequently affected, and these patients frequently have fatty livers, hepatic steatosis. Kidneys affected often, usually the renal tubules, so you can have renal tubular acidosis. In one disorder, you can have nephrotic syndrome, and I'll talk about that in a little bit. The patients often have endocrinopathies, particularly diabetes. Blood is typically not affected, but there is a rare disorder called Pearson syndrome, which is an anemia that affects young infants, and it's a sideroblastic anemia. They often have, patients have hearing loss, sensory neural hearing loss. It's a problem of the cochlea, the hearing apparatus. So it's important to note that they have hearing problems, and because it's a cochlear problem, it can often be treated when they have very severe deafness with a cochlear implant. And we're increasingly seeing gastrointestinal problems in these patients. They often have gastrointestinal dysmotility, often have trouble swallowing, gastroparesis causing early satiety, or intestinal problems with diarrhea or constipation. So I'm going to talk about these two genomes and how they cause diseases. I'll talk first about mitochondrial DNA mutations, and I'll talk about some of the classic syndromes that we see with these mitochondrial DNA mutations. This is that patient I showed you in the very first slide. This, in a headshot, this is her. So you can see again her ptosis. And what else do you see about her? She's standing next to her mother and her son. Often these patients are short and thin, especially if they have childhood onset forms. You'll notice, obviously, she looks abnormal. She's affected, but the other two, the mother and her son, are healthy. This is a disease that, although it's due to mitochondrial DNA, is typically sporadic, and it's called Leigh Syndrome, and it's characterized by onset usually before age 20, ptosis and ophthalmoparesis, and a pigmentary retinopathy. In addition, all these patients have ataxia, elevated systemic heart blocks. Okay. So that's Leigh syndrome. That's defined by Dr. Rowland, who was our chairman of neurology many years ago. This is another disorder that's one of the classic mitochondrial disorders, MRF, which stands for, we have a lot of acronyms in mitochondrial diseases. So this one stands for myoclonus, epilepsy, and ragged red fibrosis. It's hard to show myoclonus and epilepsy in a picture, so I'm showing a patient with another feature of this condition. Here you can see he has lipomas under his chin and over his shoulders. These patients often have multiple lipomas. And so it's a disease but characterized clinically by this myoclonus epilepsy and ataxia, and they often have these ragged red fibers, but also always have ragged red fibers. They may have other manifestations besides the lipomas. Okay. This is a disorder. This is the most common presentation of mitochondrial disease in children. This is called Leigh Syndrome. Are there any pediatricians in the audience? So Leigh Syndrome, it's a syndrome. There are many causes. There are over 50 different genetically distinct causes of Leigh Syndrome. But what characterizes them is, first, clinically, they have psychomotor regression or retardation, typically in early childhood, often in infancy. And the other feature is these lesions in the basal ganglia. Here you can see this T2 brain MRI. They often have lesions, they'll have lesions in the basal ganglia or brainstem. So deep gray matter structures are degenerating, and they develop a necrotic appearance. It's Neil days it was diagnosed pathologically, but now, of course, with MRI imaging, we can see the lesions very clearly. Sometimes in these families where there's a proband with Leigh syndrome, there'll be other relatives in the family who are less severely affected, and specifically those relatives are in the maternal lineage. And they may have this other condition called neuropathy, ataxia retinitis pigmentosa. And here's the fundus image of a patient initially at baseline. There are a few black spots here, but you can see that over 12 years, there's a progression of this pigmentary degeneration of the retina. So it's characterized by retinitis pigmentosa, peripheral neuropathy, and ATAX. So this and MRF and MELAS, another disorder, are maternally inherited. MELAS stands for mitochondrial encephalomyopathy, lactic acidosis, and stroke-like episodes. And the clinical hallmark is atypical strokes. I remember when Dr. Pavlakis was a pediatric neurologist here at Columbia, wrote the paper with Drs. DeVivo, DeMaro, and Rowland. They argued about what to call this syndrome. They said, well, clearly they had this mitochondrial disease because they had ragged red fibers and lactic acidosis. They had encephalomyopathy due to these recurrent strokes that cause seizures and dementia. But they argued whether to call these strokes or stroke-like episodes. Dr. Rowland said there's strokes. They often occur in the posterior part of the brain, causing hemianopsia, loss of vision in one visual field, or if they were bilateral, cortical blindness. But Dr. Pavlakis pointed out that they were atypical. Why? One is, as it says here, they occurred in young people, usually. Not always, but they often occurred in patients before age 40. And the other reason is that they didn't conform to large vessel territories. It doesn't look like a clot in a large vessel of the brain. These stroke-like episodes usually affect the cortex, so they look different. And they are different. They're metabolic strokes. We still don't know what causes the strokes or how the mitochondrial DNA mutation causes these particular events, but they're different from your typical vascular strokes due to clots. So Pavlakis won in the end, and they called it stroke-like episodes. Okay, so there were these clinical syndromes that were recognizable based on their clinical features. Hirano syndrome with ptosis ophthalmoparesis and ataxia and pigmentary retinopathy. MRF with myoclonus epilepsy with ragged red fibers. MELAS also had ragged red fibers, but they had these unusual stroke-like episodes. And then these families with proband with Leigh syndrome and others with NARP. And then there's another disease called Leber's hereditary optic neuropathy, which just affects the eyes, causing loss of vision typically in one eye, and a few weeks later, loss of vision in the second eye, usually in young people in their early 20s. So we learned that these disorders are all due to mitochondrial DNA mutations. And we first discovered these mutations, the field did, in 1988, when Doug Wallace's group identified the first point mutation in mitochondrial DNA that caused Leber's hereditary optic neuropathy, and a group in England discovered the first deletion mutations. And that same year, Doctors DiMauro, Zeviani, and Rowland pointed out those deletions occurred in patients with Leigh syndrome and PEO, obviously PEO. But in the years since 1988, we've now learned about more than 270 different point mutations of mitochondrial DNA. So what have we learned from these mutations? Well, we've learned that a different kind of genetics from what you're typically used to. This is not, obviously not nuclear DNA. It's inherited in a different way. It's inherited maternally, right? Mitochondrial DNA is exclusively, almost exclusively inherited maternally. There's only one case in the literature of a man who inherited mitochondrial DNA, part of his DNA from his father. And across most species, mitochondrial DNA is transmitted maternally. There are a few exceptions. I think mussels and redwood trees have maternal inheritance, but everything else. This mitochondrial DNA is present in hundreds or thousands of copies per cell. So therefore, you can have variable amounts of proportions of this mutation. And that's a concept known as heteroplasmy. So if you have a high level or high proportion of mutation, you're more likely to be more severely affected. Whereas if you have a low percentage of mutation, you're going to be less affected or even asymptomatic. We noted that the distribution of the mutation can vary from tissue to tissue. And I'll give you a good example of that. I mentioned that there is a rare blood disease that affects infants called Pearson syndrome. Those patients have the deletion mutation that also causes Leigh syndrome. They have it in their bone marrow, and it's present in abundant amounts in the bone marrow, and it affects the development of the blood cells, and these patients develop red blood cells with these sideroblasts, iron deposits. Well, what happens over time is sometimes these patients can have severe anemia and require transfusions, and sometimes they die of severe refractory anemia. But sometimes these patients, with support of transfusions, can survive, and they gradually lose that anemia. Why? Because there's a kind of Darwinian selection that goes on in the bone marrow. The cells with normal mitochondrial DNA repopulate the bone marrow, and they lose the deletion from the bone marrow. What happens over time then in these patients? Over the next few years, they often are healthy, but then later in childhood or early adolescence or even adulthood, they start to develop drooping of the eyelids and ophthalmoparesis, the features of Leigh syndrome, because those deletion mutations accumulate in other tissues like muscle, in post-mitotic tissues. So in the bone marrow, you could select cells with normal mitochondrial DNA, but you can't do that in muscle because they don't replicate very much. So those tissues that are post-mitotic accumulate deletion, and the phenotype changes because of the segregation of the mutation in different tissues. And then the threshold effect, as I told you, brain and muscle require lots of energy, so the threshold for symptoms in those tissues is relatively low. Most tissues are frequently. So this is and to highlight how heteroplasmy matters. This is that those two disorders I mentioned before, where there's a severe maternally inherited Leigh syndrome that affects young children, and then there are other people in the same family who have the NARP syndrome, and there may be others who are asymptomatic, yet they all in the maternal lineage, and yet they all carry the same mutation. They have the same one of these two mutations in the ATPase gene. But the mutation load varies. In the patients with NARP, they may have 70 to 90% mutation. In patients with Leigh syndrome, they have over 90% mutation. In patients with less than 70% mutation are often asymptomatic. Same mutation, multiple presentations within the family. That's typical. So here's what we often see with these families. We see the proband here. In this case, this was a young child who had started with a exercise intolerance, inability to run because of myopathy. She then developed stroke-like episodes around age 5, unfortunately, and then progressed to develop cardiomyopathy and died at age 10. So she had the Milos syndrome with the stroke-like episodes, but her brother, who had some mild mental retardation, seizures, was less affected. The mother was completely asymptomatic, and a maternal uncle was mildly affected with some ptosis and ophthalmoparesis. This is typical. You have to dig carefully into the family history to find these other people who are oligosymptomatic and in the maternal lineage. So it's important when you, as genetic counselors, when you take family histories to dig deeply into the maternal lineage to look for these clues of maternal inheritance. And then if you do the mutation analysis, in this case it was the most common amino loss mutation, the 3243 mutation. You'll see that the proband here has high levels of mutation in blood and muscle. And the others who are less severely affected have lower levels of the mutation in these tissues. Interestingly, this, you can see here, this person here, who's asymptomatic, has no detectable mutation in blood. Sometimes it happens. But if you look at other tissues, you'll see that, that there's a detectable mutation in muscle, for example. And one thing we've learned with this particular mutation and other mitochondrial DNA mutations is that the mutation load is often very high in the urine. So we often ask patients to give us a urine sample, spin it down, and measure or look for mutations in urine. We learned, this is a little out of date, there's actually a newer paper that just came out in the Annals of Neurology that showed that in working age individuals, the frequency of symptomatic mitochondrial disease is about 1 in 5,000. This is an older paper, but right now it's about estimated to be 1 in 5,000, so it's not uncommon. And if you look for presence of these common mitochondrial DNA mutations in the population, and in this case they looked at umbilical cord blood of healthy individuals, and they found that 1 in 200 people carries a known pathogenic mitochondrial DNA mutation. So there are a lot of carriers out there, but obviously not all of them are affected, and it's because their level of heteroplasmy is below the 70% critical threshold for developing disease. So these mitochondrial DNA mutations are not uncommon. So I'm just going to digress a little bit and say that we're working on strategies to try to treat these mitochondrial DNA mutations. We, as in the field is. So one of the advantages we have in mitochondrial DNA mutations is that there's a mixture of wild type and mutant mitochondrial DNA. And I give you the example of Pearson syndrome, where if you get rid of the mutated mitochondrial DNA, you can get rid of one of the symptoms, anemia. Well, what if we could do that in patients? What if we could get rid of that mutated mitochondrial DNA and let the wild type mitochondrial DNA repopulate the cells? Well, theoretically, that's possible. In the case of NARP, this particular mutation, this 8993T, the G mutation, it creates a unique restriction site for a restriction enzyme, SMA1. So if you could insert that SMA1 enzyme and put a mitochondrial targeting signal so that it goes into the mitochondria, you could cut the mutated mitochondrial DNA, it'll be degraded, and it'll disappear, in theory. And it's been done by a group in Japan, by Tanaka and colleagues, and it worked in cultured cells. But not all of these mutations create unique restriction enzyme sites, so this is not a strategy that's going to work very much unless you can custom design enzymes. And you can do that now with zinc fingers. And this is a group in Cambridge, England, where they designed zinc fingers that bound to mutated mitochondrial DNA, and they were able to increase the level of the wild-type mitochondrial DNA with these zinc fingers. They have to design two because the nuclease has to line up on both sides of the DNA, and to cut the DNA, and they were able to shift the heteroplasm. That's terrific in cultured cells. And another approach is to use something called TALENs, this transcriptional activator like endonucleases. So it's very similar to the zinc fingers. They're proteins that can be custom designed to bind to specific sequences of DNA. And this is work from Carlos Moraes' group in Miami, where they used TALENs to cut one of these deletion mutations and eliminate cells and a mixed population of mutated and normal mitochondrial DNA. Here you can see the shift in heteroplasmy, the increase in the wild-type mitochondrial DNA in the cells that express both of these TALENs. Well, that's great, but this is still science fiction because we can't do this. We can't deliver these talents and zinc figures to cells yet. Gene therapy has to be optimized. And also these talents still need to be optimized. But what if we could shift the heteroplasmid from one generation to the next? What if we could prevent the transmission of mitochondrial DNA? That would be a good thing, I would think, right? Is there a way to do that? Yes. Maybe you heard about 3-parent embryos. It was in the news. It was a hot subject for a while. Well, this has been really pioneered by the group in Newcastle, England, Doug Turnbull, Mary Herbert, and colleagues. And they were the first to show that you could take embryos, and these were embryos with abnormal numbers of nuclei, and you could transfer the pronucleus from one embryo and then put it in the cytoplasm of another embryo after you nucleate it, and you will end up with a cell with basically 3 parents. You'll have the mother and father who contribute nuclear DNA, and you have the mitochondrial DNA donor. You can do that using pronuclei. And then you then implant this embryo into a woman, and you can develop a child, hopefully, which will be free of the original mother's mitochondrial DNA mutation. Another approach that has been used by my colleagues, Dieter Egli, here at Department of Pediatrics, and Mark Sauer, is to do the nuclear transfer at the egg level, at the oocyte level. So you take 2 oocytes. You have a woman with a mitochondrial DNA mutation carries it in her oocytes, and a healthy mitochondrial donor who donates her eggs. You take the nucleus out of the oocyte with the mitochondrial DNA mutation, and you transfer that chromosomal spindle into the cytoplasm of a healthy oocyte. And you then do IVF, and you could theoretically create a child who has the nuclear DNA of both biological parents and mitochondrial DNA of a donor. So 3 parents, right? Think about it. And I talked about that in this room a few months ago with the others. This approach has been used successfully in primates. There's a couple of monkeys that were born. They had these are macaque monkeys. They were generated after chromosomal spindle in the oocyte level, and they used 2 subtypes of macaque monkeys with different mitochondrial DNA haplotypes, and they were born. This paper was published in 2009, I believe. So these monkeys are now more than 6 years old and apparently doing quite well. So you can manipulate the nuclear chromosome, do these transfers, and produce healthy primates. So in the UK, actually, Parliament approved that. That was why it was in the news recently. And they are just working with the regulatory agencies in the UK to develop guidelines on how to do this in specialized clinics that have the expertise in IVF and in mitochondrial disease. And the expectation is that they will do their first mitochondrial replacement therapy later this year. So the first child born of this with this procedure will probably be born in 2016. Here in the United States, we're lagging behind. A number of us have been interested in trying to do it here. And we've approached the Food and Drug Administration has been approached. They have asked the Institute of Medicine to investigate this. So the Institute of Medicine is doing a study, which is going to take more than a year. So we're not going to. So this is going to be done in the UK before it's going to be done here. But nevertheless, it's something that we're working on here at Columbia with Dieter Egli, as I mentioned, and Mark Sauer, who's the director of the IVF clinic here at Columbia. All right, so I talked quite a bit about mitochondrial DNA, which is a unique aspect of that organelle. I'm going to switch now to the nuclear genome, which obviously most of you know more about. It's obviously quite huge. It codes 20,000 genes as opposed to the 13 genes encoded by mitochondrial DNA. But a surprising number of these nuclear genes are required for mitochondrial functions, about 1,600. So almost 10% of these nuclear genes are required for mitochondrial function. So that means that quite a few nuclear gene defects, maybe 8% or so, will be mitochondrial diseases due to nuclear gene defects. Now with next generation sequencing, new nuclear genes are being associated with human diseases, about 1 paper a month. It's quite frequent. So I mentioned that mitochondria, obviously are important for making ATP, but they have other functions. They have to maintain their own mitochondrial DNA, and that's dependent on nuclear gene factors. They have to transcribe that mitochondrial DNA into RNA. They have to translate it into proteins. They have to assemble these complexes, these multi-subunit complexes. They have to maintain their lipid membranes. So they're quite busy places. I told you they look like these kidney beans shaped structures on electron microscopy, but they're very dynamic. They move. Here you can see mitochondria. So let's turn this on again. And they move along microtubular rails. And you'll see here, over here, if you look at this mitochondria, it fused with the other one, and then it's split up. So they're constantly moving, fusing, dividing, very dynamic. They're also critical for apoptosis and many other cellular functions. So, mitochondrial DNA mutations can only cause defects of mitochondrial protein synthesis through mutations in tRNAs or ribosomal RNAs, or they can affect these protein coding genes that are components of the respiratory chain. But nuclear DNA defects can affect many more functions of mitochondria. In addition to their direct hits, just like in mitochondrial DNA, you can affect these subunits. The blue ones here are covered by nuclear DNA. So you can have monoenzymopathies due to these structural subunit mutations. You can have similar monoenzymopathies due to assembly factors that are required by these enzyme complexes. And these are all encoded by nuclear DNA. So there are 2 levels of hits on these respiratory chain enzyme complexes. You can affect, just like mitochondrial DNA affects protein synthesis, nuclear gene defects can affect mitochondrial translation. And in many other ways besides through tRNAs and ribosomal RNA defects, through factors required for elongation and termination and so on of translation. Okay, but there are new disease mechanisms that are constantly being identified. This slide's out of date, but even 5 years ago, there were over 100 different genes that were associated with mitochondrial diseases. And most of these were nuclear genes, and now that number is closer to 200. And as I mentioned, it's growing by about 1 gene per month. One of the important components of the respiratory chain that's often overlooked is this thing called coenzyme Q10. Probably all heard about it. You've seen it in your local drugstores or Costco. Well, what is coenzyme Q10? It's this molecule that shuttles electrons from complexes 1 and 2 to complex 3. And it's called Q10 because there are 10 isoprenyl groups here on the side. So it has this very long tail and this head structure which carries electrons. And coenzyme Q10 is synthesized within mitochondria. These are, here you can see the biosynthetic pathway, and they require proteins that are encoded in nuclear DNA to make the final product, coenzyme Q10. So there have been a variety of phenotypes associated with CoQ10 deficiency, often in early childhood onset encephalomyopathy, brain and muscle disease, like many of these mitochondrial diseases, and infantile multisystemic disease with brain involvement, and kidney disease, often nephrotic syndrome. So this is one of the unusual mitochondrial diseases that causes nephrotic syndrome, protein loss through the urine. And they often have cerebellar ataxia. Well, we studied one family with this infantile onset multisystemic disease. The proband was a young child from Italy. He was 33 months old. At age 1 year, he developed the nephrotic syndrome with proteinuria, became hypotonic, and developed these stroke-like episodes very similar to immune loss, and cyclomotor delay and optic atrophy. So he had a complex multisystemic disease. when we looked at his muscle biopsy and measured activities of these enzymes, complex 1 plus 3 and 2 plus 3, in combination, they were decreased. But individual activities of complex 1 and 3 were normal. It's because they were lacking coenzyme Q10, the shuttle between these enzyme complexes. And it was very low, Q10 was very low in muscle and fibroblasts. And his sister was developing proteinuria at age 9 months. And we started giving both of them coenzyme Q10. Unfortunately, the older boy had this renal failure and required a renal transplant. When he was given coenzyme Q10, he started regaining developmental milestones and he improved. And the younger girl's proteinuria improved, and she has not developed any neurological problems. So here's a treatable mitochondrial disease. They can't make CoQ10, so you can give them CoQ, which is great. We did homozygosity mapping, and we found this homozygous mutation because the parents were consanguineous. We found this homozygous mutation in this gene called CoQ2, which is responsible for condensing the para-hydroxybenzoic head to the decaprenyl tail. And now we know about mutations in 8 different coenzyme Q biosynthetic genes. So these defects cause primary CoQ10 deficiency. They can't make it, so there's a primary defect in coenzyme Q10 biosynthesis. And these patients typically present with an infantile multisystemic disease. Again, it's a brain disease, often with nephrotic syndrome or kidney involvement. And there's a milder form with just ataxia, or sometimes ataxia and seizures. And that's due to a kinase which regulates CoQ biosynthesis. And then there are these other disorders where there's a primary defect in another gene unrelated to CoQ10 biosynthesis, but they have secondary CoQ10 deficiency. And Katerina Quincy in our lab is working on the mechanisms of that. Okay. There are now defects of mitochondrial dynamics, the fusion, fission, and movement of mitochondria that can be associated with diseases. The most common form of inherited neuropathy, Charcot-Marie-Tooth type 2A, is due to mutations in this protein called mitofusin 2, which is required for fusion of mitochondria and then a fusion of the outer membranes. And this dominant form of optic atrophy, most common autosomal dominant form of optic neuropathy, is due to mutations in OPA1, which is required for fusion of the inner mitochondrial membrane. There's even a defect of mitochondrial fission, which causes a multisystemic disease. As I mentioned before, mitochondrial DNA depends on nuclear gene factors to replicate the repair itself. And so if you can have primary defects in the nuclear DNA that cause instability of mitochondrial DNA, and that causes multiple deletions. I'm in trouble with the here, but you can have either multiple deletions of mitochondrial DNA, or you can have a lack of mitochondrial DNA, which is called depletion, or you can even have a combination, as in this disease called MNGIE, another acronym. MNGIE stands for mitochondrial NeuroGastrointestinal Encephalomyopathy. It's a multi-systemic disease, obviously. And I'll just tell you briefly a story of how we went from patients to identify their genetic defects and identify the pathogenesis of the disease, and now we're going back to patients with therapy. And this is what we all are trying to do with precision medicine these days: identify mutations and identify their disease pathomechanisms, and hopefully come back to the patients with meaningful therapies specific for their disease. So this is an example here. This MNGIE, this is a patient with that disease. He has droopy eyelids too, and his eyes are looking in different directions. He has ophthalmal paresis. And he has this severe leukoencephalopathy. His white matter is very abnormal here. And the middle panel is the nerve conduction study, which shows his nerves transmit electrical signals very slowly because of his peripheral neuron. So those are the major clinical features: the ptosis ophthalmal paresis, the gastrointestinal dysmotility, which causes the patients to become very cachectic, and then they have peripheral neuropathy, leukoencephalopathy, and then mitochondrial abnormalities, including deletions and depletion, and ragged red fibers in muscle. Now, this disease is devastating. It typically begins in the late teenage years, and the average lifespan of these patients is about 38 years. They suffer for about 2 decades with this disease. It's a devastating disease. Years ago, we identified the causative gene back when we had to do linkage for next generation sequencing was around. And we found the causative gene is TYMP, which encodes an enzyme called thymidine phosphorylase that's not in the mitochondria. It sits in the cytoplasm, but it's required to degrade these nucleosides like thymidine, deoxyuridine, into the base thymine and the sugar ribose. So we measured the thymidine phosphorylase activity of myself and a bunch of postdocs in the lab, and this is what the normal activity is in blood. And we found that all of the patients had very low activity, less than 10%. So clearly these mutations were causing loss of function, causing the disease. We also found that the mutation carriers, the parents of these patients who had one mutation, had 30 to 50% enzyme activity and were completely healthy. So it showed that you only need partial enzyme activity to be normal. And then we found a small group, 3 patients, who had a late onset disease that started in their 40s. And they were alive in their late 50s and early 60s. And they had partial enzyme deficiency. They had about 10 to 15% enzyme activity. So you can see a clear correlation between enzyme activity and severity of the disease. Obviously, the more severe the defect, the more severe the biochemical defect, the more severe the phenotype. Nice. So how does this enzyme deficiency cause the disease, and how does it cause mitochondrial DNA instability? Well, I told you this enzyme degrades these nucleosides, and they accumulate in the blood and tissues of patients. And these nucleosides are incorporated by the mitochondria, and they go through this salvage pathway, they're phosphorylated to make the nucleotide triphosphates, which are required to make mitochondrial DNA. So there's an imbalance of these substrates required to make mitochondrial DNA, and that causes damage to the mitochondrial DNA and the deletions and depletion of mitochondria. So we figured that out. We made a mouse model which replicated many of the features of this disease. We made a TPUP doppel knockout mouse, which developed this kyphosis, and they had a local encephalopathy, mild, compared to the patients. And they have partial deficiency of mitochondrial complex 1 and partial depletion of mitochondrial DNA in the brain. So they recapitulates many of the features of this disease. We then stressed the mice by giving them thymidine and deoxyuridine, and the phenotype got worse. They died early, and they developed more severe leukoencephalopathy, brain disease. So that confirmed that the toxic metabolites are the cause of this disease, thymidine and deoxyuridine. Well, while we were studying this mouse model and characterizing the disease, we were treating patients at the same time, of course, and unfortunately, they were getting worse, and several of them had died with the disease, so we felt compelled to do something for them. And this is a young woman who came to us from Italy with the disease. She was 30 years old at the time. She's a pharmacist, so she understood the disease very well, and she was pretty severely affected. She was 5'2" in height and weighed just 25 kilograms or 55 pounds. She was very emaciated. She had such severe gastrointestinal dismotility, she was fed intravenously, 2,400 calories a day. And she could only take about 400 calories a day by mouth. She had severe abdominal pain and bouts of pseudo-obstruction of her intestines and diarrhea every week. And she had weakness from her muscle disease. She had ragged red fibers, and she had weakness of her proximal lips. So she had trouble lifting her head from a pillow and climbing upstairs or getting up from a chair. She couldn't run or jump. So she was quite weak. And she had a mild neuropathy with tingling in her feet and absent tender reflexes. So we reasoned that if we could restore enzyme activity or get rid of these toxic metabolites, we could treat the disease. So with Ramon Martí in Spain, we infused platelets into patients because it contains abundant amounts of TP enzyme. And we showed that, yes, you could restore enzyme activity through platelets and reduce these nucleoside toxins from the blood. And it worked. But it only worked temporarily because platelets turn over very fast. But it was just a proof of principle. So a more permanent solution is bone marrow transplant. Permanently restore the enzymes and have cells in the circulation that can eliminate these toxic metabolites. Well, in this woman, She had an ideal donor. Her brother was HLA identical, a perfect match. And he donated his cells. She had a successful engraftment initially at about a month after the transplant. She had about 60% donor cells in her circulation, and she started to lose the transplant. We stopped the immunosuppression. And by a year out, she had 100% donor cells in her circulation, which was great. The transplant was successful. And with that transplant and engraftment, she had restoration of the enzyme activity in her blood. Here's the enzyme activity. You can see it was undetectable before the transplant. Your levels that are in the low normal range. And that enzyme was able to eliminate these toxic metabolites, thymine, deoxyuridine, to levels which are virtually undetectable at this point. Now it's almost 10 years after the transplant. Slide's a lot out of date, but she's improved clinically. Her gastrointestinal problems have improved. She no longer has these bouts of pseudo-obstruction, and she's stopped her intravenous feeding. She's eating by mouth now, about 3,000 calories a day. She's stronger. She's able to run. She likes to visit us once a year in New York and runs up and down the hallway and jumps up and down. And now she likes to go sightseeing in New York. She was too weak. She does that with the money. And her neuropathy has improved subjectively. Pins and needles have gone away. Objectively, she has reflexes, tendon reflexes in her nerve conductions. So that's great. There are other disorders that affect these nucleotide pools, and they cause mitochondrial DNA instability. And there are also defects in the mitochondrial DNA replication machinery, particularly this enzyme, polymerase gamma, which is responsible for mitochondrial DNA replication. And mutations in in pol gamma are probably the single most common nuclear gene group of nuclear genes, or at least single most common nuclear gene that's causes mitochondrial diseases. And it presents in many different ways. It can present either as autosomal dominant or recessive ophthalmoplegia. You can have a multisystemic disease like sensory ataxic neuropathy, dysarthriate ophthalmoplegia, or SANDO. Or we can even have children affected with a severe brain and liver disease called Alpers-Huttenlocher syndrome. It presents with severe liver disease, often causing liver failure and refractory seizures, and unfortunately these patients die often early in childhood. But this is quite heterogeneous. Okay, so I told you about a lot of different diseases. They're quite heterogeneous. I think that the heterogeneity of mitochondrial diseases is going to get even more complicated because we find new causes of mitochondrial diseases. So as I said, we're finding a new gene every month. So I just wanted to leave you with a few clues in diagnosing these patients. First, these diseases are often multisystemic, but I told you they affect different organs in different ways. And specifically, they affect the brain and muscle frequently. But if you see that in combination with a fatty liver, or hypertrophic cardiomyopathy, or hearing loss, think of a mitochondrial disease. Those are the red flags you should look for. Ptosis, ophthalmoparesis, hearing loss, diabetes, fatty liver, cardiomyopathy. These are the things that we often ask in family history. Don't forget that there can occasionally be tissue-specific disorders like the liver's optic neuropathy, so they affect just one organ. Family history is important. Obviously, the nuclear gene defects can have autosomal dominant, recessive, or even X-linked inheritance. But think about maternal inheritance and dig deeply into the maternal lineage, asking about patients who might be, or relatives who might be oligosymptomatic. Laboratory clues. These patients often have elevated lactate in the blood, particularly children. It's not so common in adults, but in children we often see elevated lactate. These patients often have exercise intolerance, so if you do exercise testing, you'll see that they have early anaerobic threshold, that they can't exercise like a normal person. And the neuroradiology MRIs can often show particular lesions like immune loss or Leigh Syndrome. Molecular genetics. So it's we're in an era of transition. In the old days, we obviously had great difficulty sequencing, but now with next generation sequencing, we can do panels or whole exome sequencing to find these mutations. And mitochondrial DNA is particularly good for next generation sequencing because we can sequence it 1,000 or 7,000 fold depending on the technique, and we can pick up low levels of mutation, pathogenic mutations. All right. So, and then when we're uncertain, when we get these variants of uncertain significance, muscle biopsies can be very helpful in allowing us to look at the morphology, look for ragged red fibers, and look for biochemical defects. And of course, we can do functional studies in cultured fibroblasts from patients. We have a lot of tools now to study mitochondria, so it's an area where we can do a lot more than just sequence, and we can do functional studies to prove the pathogenicity of innate